From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on August 21st, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 105th episode of Matters Microbial. Many thanks to every listener, viewer, guest, and supporter. It really matters. First, I wanted to thank Jerry Jackson for emailing me about compost microbiology and suggesting it might be an interesting session for the future. It's certainly one that many homeowners would find interesting, as do I. Thank you for the kind words and suggestion. I'll keep my eyes out for a good suspect. Next, some sad news. Dr. Moselio Schechter passed away recently at the age of 97. Elio was an important part of my developing into a better microbiologist and communicator of science. Honestly, Elio was so sharp, wise, and knowledgeable about the microbial world, and such a kind and helpful human being. Here is a photograph of Elio and yours truly at an ASM microbe meeting a few years ago. My wife, Dr. Jennifer Quinn, helped me honor Elio's memory, example, and impact with 2 Lux portraits painted using luminous bacteria. The first is an excellent likeness. The second shows his impish side, which always delighted me no end. I'll put some links up about Elio in the show notes. The world is less bright without his microbial fire and his wisdom. I'll miss him a great deal. As you know by now, I'm fascinated by bacterial bioluminescence. Here is a Lux portrait of me I took just a few days ago, illuminated by a flask of glowing bacteria. One thing that's vital for scientists to accept is when their wonderful observations or hypotheses do not pan out. I'm wrong, or I don't know, are not indictments, but an opportunity to learn, grow, and think more deeply. I have long been interested in marine microbes that digest the agar in petri dishes, my famous pitters. So imagine our surprise when a student found what appeared to be pitters on a plate that had not been inoculated. You can see the pits around what looked like colonies. We were very excited. But sadly, nothing grew on restreaking onto the same medium, let alone forming pits. So we really aren't sure what's going on. But honestly, looking carefully at plates has also revealed very wonderful results as well. Observation matters, but must be followed up by confirmation, and sadly, repetition. For example, here is a dilution plate from one of my stressed cultures of luminous bacteria. You can see bright, dim, and dimmer colonies easily. My student Chiana noted that some of the dim colonies looked fuzzy, while others were more defined, as seen here with the arrows. We were very excited. Yet, when Chiana restreaked the different fuzzy and non-fuzzy dims, they looked the same. So an exciting initial observation had been defeated by a confirmational experiment. But that is what science is all about. Repetition and confirmation. It's what I like to call the in research. It's vital. We look around us today and we see why. By the way, that stressed culture created this lovely movie. The different levels of brightness rather looks like a galaxy. Microbiology and art meet. I've also been interested in marine microbiology for decades, but I was an in-lab sort of researcher. Other folks go out on exploratory cruises, have deep-sea submersibles, and they find and see wonderful things, like creatures in the deep ocean that use bacteria to provide them with carbon in a process called chemoautotrophy. Marine microbiology plus symbiosis equals a wonderful combination for Doc Martin. So I'm very pleased to introduce Dr. Roxanne Beinart of the University of Rhode Island to talk about the work her research group has done on some of these marine symbioses using biochemical reactions to fix carbon dioxide into organic molecules. Welcome to the Quality Quorum for today, Roxanne. It's a pleasure to meet you.
Thanks so much for having me, Mark. I'm happy to be here.
I am so glad you agreed to be here because the things that you study are of great interest to me. And I want to tell listeners and viewers that there is going to be a link to Roxanne's websites, and you must look at her research lab website, which is just fabulous.
Thank you.
Roxanne, would you mind kind of telling everyone here a little bit about what chemoautotrophy is about? Because it sounds like a mouthful to people who haven't been through our courses.
Sure. Another way to say chemoautotrophy is chemosynthesis. And that sometimes is an easier word for people to understand because it can be very analogous to a process that people are often more familiar with, which is photosynthesis. So photosynthesis is the process of using light energy to turn carbon dioxide gas into sugars that obviously then fuel plant growth and support food webs on Earth and in the sunlit ocean. But there is another process on Earth that allows organisms to take carbon dioxide and turn it into sugar. But instead of being fueled by the sun, it's fueled by chemical energy. And so that's where the chemosynthesis comes from. And so my lab works on chemosynthetic microbes that live at the very bottom of the ocean where these chemical energy sources are abundant. And they do this in partnership with animal hosts. And I can certainly talk more about that now or later if you want to hold on.
Oh, I am happy to go where, but I want to remind listeners and viewers again that the work that I do is just at the bench. Sometimes I'll go to the seashore to some of these hydrogen sulfide seeps that we have locally. But you, on the other hand, go on cruises. Would you mind telling everyone what that means? Absolutely.
So you use the word cruise, which is a word that we oceanographers use to describe a seagoing expedition, where we're taking a large oceanographic vessel, something that's very large, out to the middle of the ocean to reach the parts, areas of the ocean that are otherwise inaccessible. So we call it a cruise, but it's a working cruise, not a leisure cruise. It can be confusing for many people to hear the word cruise. So expedition is another way of thinking about it. And so, yes, we are able to go out on these large research vessels. The research vessels are owned and operated by the U.S. government, but universities are the ones that are actually facilitating them. And so we have access to the ocean on these vessels. On these vessels, when we want to study the deep sea, we often also need to bring with us deep submergence vehicles or submersibles. And that allows us to actually dive deep into the ocean to actually access the sites that I work on. So I am typically going out to study deep sea hydrothermal vents and cold seeps. And these are areas of the seafloor that I can talk more in detail about what they're like in a second, but they can be kilometers deep in the ocean. And so we obviously cannot scuba dive or swim to them. And so if we want to reach them in order to collect samples for our studies, we need to use these deep submergence vehicles. One type of deep submergence vehicle is a submersible, like a submarine, people might be more familiar with, where people are actually journeying themselves in the vehicle to the very bottom of the ocean. And I actually have not really worked much with submersibles, although there are some really impressive scientific submersibles in the US, namely the HOV Alvin, which is the sort of most well-known science submersible in the US. But I primarily work with another type of vehicle where we are sending a remotely operated vehicle, an ROV, to the seafloor that can act as our eyes and hands. And so these are large vehicles, the size often of a car or a minivan, that are sent on a cable to the seafloor and controlled from engineers and pilots who are on the ship. And this vehicle has lights, and it has cameras, and it has manipulator hands that actually can grab samples for us. And so you can see here in this photo of me with one of these remotely operated vehicles, where they can be quite large. This is the RV when it's on the ship, but this is deployed over the side of the ship and sent to the seafloor, where it can then collect what we need to do our studies and then return to the ship with the samples.
It's like a very, very expensive video game / drone.
Yes. And it's true that the pilots and operators and engineers who drive these vehicles, it can be very much like playing a video game in some ways. They have, often 10 different screens they're looking at all at one time. They have even controllers that are very similar to what you would see in a video game setup. And it takes a lot of spatial awareness that might be similar to someone who's, if you're interested in video games, this might be a career that you would consider, because you need that kind of spatial awareness and hand-eye coordination to be able to operate these vehicles. And the other thing sorry.
No, go right ahead.
The other thing I should note too is that we as scientists are not directly operating these vehicles. There's whole teams of professionals that we work with, engineers and pilots, whose job it is to maintain these vehicles and to operate these vehicles. And so we work with them to direct the operations to get the samples we need, but I don't get to drive the ROV. So unfortunately.
No, I like the honest aspect of it. Now, again, how long are your cruises? Pardon me, your research expeditions?
Great question. The oceanographers can go to sea for anywhere from a single day to months. My research expeditions or cruises have been on the longer side, and that's typically because of the locations that I tend to study. For example, one of my study systems is around Tonga, which is, in the South Pacific. So it's a long way away from here. And so the longest research cruise I've been on has been 49 days at sea, but they are more commonly in the 25 to 30 days. So, between 3 and 4 weeks is fairly common.
We've had other guests on the podcast that have talked a little bit about this experience. And I just wanted to emphasize your personal experience because I'm getting ready to teach a class. Mm-hmm. I run into many students who think that doing microbiology is essentially just looking at plates in the lab, and there is that. But there's if you'll forgive me, and I know the reality isn't quite as gleaming as I portray it, it's a lot more adventurous.
Absolutely.
To be out there. And again, I'm well aware that the food and the seasick and all the rest, I have it, but it's still a lot different than people think. And I like listeners and viewers to understand that. So if you don't mind, I think a really good idea is to talk about one of your study systems that you look at, perhaps your snails, mm-hmm. and lead us into what's going on.
Absolutely. So one of the main topics of research in my lab has been focused on hydrothermal vent systems that are in the area of Tonga in the southern Pacific Ocean. And so at vents in this part of the world, the dominant chemosynthetic symbioses are partnerships between mollusks, so that's snails and mussels, and bacteria. And so in all of these cases, these animals are relying on the microbes that they partner with to provide their primary nutrition. So what that means is that the chemosynthetic microbes live inside the tissues of these animals, and then are using this process of chemosynthesis to take the chemical energy coming from the hydrothermal vents, and turning that into energy that's used for the fixation of carbon into sugars, which then feeds the host almost entirely. So these snails and mussels that I work on do still have, they have a mouth, they have a gut, so they in theory can intake food like a shallow water snail or mussel might. But what we know from a lot of studies on these types of systems previously is that most of their nutrition, most of the sort of carbon in their bodies comes from their bacterial symbionts. And so what this is like, what I always like to say is that this is like as if you and I could feed ourselves entirely by growing corn in our bellies and standing next to a window. These animals just have to sit next to these hydrothermal vents, which are like deep sea hot springs. And there's chemicals that are coming out in this fluid, and they sit there and their bacteria grow and provide them with pretty much all of the nutrition that they need.
Roxanne, I love this diagram where you're starting to talk about a little bit about how the symbiont and the vent fluid interact a little bit to fix carbon dioxide. And so some questions about that are occurred. So the first thing is, I'd like you to kind of look at that image a little bit, and I'm going to ask the following. How do the symbiotic bacteria actually enter the gill cells, which is a subject that I'm really excited about?
So as you can see in this figure here, I have an image of what's supposed to be a gill cell of one of these animals. So this is the cell of the gill of the animal. The gill is the structure that the animal uses to breathe. And inside of the gill cell, it's packed with these bacterial symbionts. We don't actually know directly how these symbionts get into the gill cell. We know that in these symbioses that the animals acquire their symbionts from the environment at some point in their development, either as a larvae or juvenile, but we don't really know when and how that occurs.
I would imagine that some of these cells can do the equivalent of phagocytosis. And I know that bacteria that invade eukaryotic cells often take advantage of that natural process. And I don't know, as you say, enough about gill cells to think if that's relevant.
Yes. Yeah. I mean, I think that's likely. And we also see, in some cases, you'll see kind of the bacterial cells on the surfaces of the gill cells, so they may, be phagocytosed into the cell. That might be one way. There is some indication from some of these types of symbioses that they may come in through other tissues too, not just the gills, and migrate somehow through the animal's body to the gills. So it may come directly through the gills and or from other tissues as well.
I mean, if you look at, for example, Wolbachia present in many insects, they tend to populate. And it makes sense in the backwards thinking that often fools us, that usually ovarian tissue seems to be highly colonized, but sometimes other tissues as well.
Right.
So I love this idea a lot. And when we look, and I don't know if they're called bacteriocytes when you do this work. That's what they call it in a lot of insect symbioses. But do you know anything about how they control the population number of the symbiotic bacteria within the cell?
So I think there is some indication that there is control from the host, where they are actually potentially killing some of the symbionts in the cell. We do see active evidence of division in the host cell, so the cells don't appear to be in some sort of state where they've stopped dividing. They do seem to be dividing still, which would mean that the host would have to control the population. One of the ways that they might do this is through the active digestion of the symbionts. And that's also how the carbon is transferred from the host animal to from the symbiont to the host animal. So if you look in this figure here, I have this sort of schematic depiction of the process that's going on, where you have hydrogen sulfide or methane or hydrogen gas being used by the bacteria. It partners that with either oxygen or nitrate, and together those two chemicals provide the energy that can be used for carbon fixation. So then they take CO2, carbon dioxide gas, turn it into sugars, which the bacteria use for their own biosynthesis and growth, but also ends up providing the nutrition of the host animal. And that's probably mostly through the whole digestion of these bacterial cells. So the animal is actually breaking down the cell, digesting it, and taking all the components and parts and using that as their nutrition instead of taking in food.
I have to tell you, you really took me back to graduate school because in the symbiosis between Cynorhizobium, Melilodi, and alfalfa, these bacteria never really enter the cytoplasm of the cells that they invade. They're enclosed in a series of plant-derived membranes called the peribacteroid membrane, and that controls what goes in and out. I love the idea of the gill cells digesting the organisms directly, which infers they have to be controlling how the bacteria are reproducing inside.
Right. Yes. So by digesting some of them, they're sort of keeping the populations down or controlling the populations, while at the same time, that's how they're getting the nutrition they need by essentially eating these bacteria that are living inside of their bodies.
Would you do me one really bizarre favor in your research if you ever can?
Sure.
I have a theory that when bacteria live inside the cytoplasm, directly exposed to it anyway, they overexpress GRO-EL and GRO-ES, the two chaperones that help proteins fold properly. I would love to know if your symbionts are doing that too.
Yeah, that's interesting. Yeah, we have a little bit of gene expression data. So we are sometimes able to work with these animals alive. We also can sometimes capture their gene expression in situ at the bottom of the ocean. And so we have done some gene expression work, but I can't remember if we looked at those particular genes, and we certainly can do that either from that old data set or in the future.
I think listeners and viewers should keep in mind, and this is something I talk about with students a lot. You look in most textbooks, and the focus is using photosynthetic or chemosynthetic processes to make sugars, and that does happen.
Mm-hmm.
But what it's also doing is making precursor metabolites that allow the organism to actually build everything organic. So that's the definition of autotrophy. And so this idea that you just mentioned, that an animal is using a similar approach to plants, and of course, that's from bacteria originally, I'm well aware. But the point is still germane, that they're able to build themselves up from CO2 and chemical energy instead of light is just amazing.
Yeah, it really is. I mean, and beyond just being able to do this, these animals can also grow quite large and quite abundant. So, for example, some of the snails I work on are very, very large, can be the size of a softball in their sort of largest stages. So if you see here in this photo of me with one of these giant snails, you can see me holding one, and you can see quite how large they are. They can be easily softball sized. And so this is a situation, again, where these animals are not eating. They're farming these bacteria inside of their tissues and yet can grow to these enormous sizes. And also into these incredible abundances of animals at the seafloor. So they can be as dense as what we see in the intertidal. If you were to go to the rocky intertidal and see these beds of mussels or other animals coating the rocks, we see the same types of abundances at the seafloor with these vent animals, and all relying on this bacterial symbiont. The other part of this that's important to note is that these are ecosystems that are normally food poor. So when you're at the bottom of the ocean, there's just not a lot of food there. And so we get these incredible sizes, these incredible abundances in a place where there's almost no food available to animals normally. And this, again, is because they are farming these bacteria.
It's so interesting to me. If you go further up Puget Sound, where there's a lot of buried lumber, you'll see here in the Puget Sound area certain amounts of mussels that are able, within their gills, to have chemosynthetic bacteria that they're indeed farming. And I'm not clear about whether or not they're intracellular in that case, but the ones that you're talking about, say with your snails, they've gotten into the tissues. Is that correct?
That's right. Yes. So they live in the gill tissues, and they actually live inside of the cells of the animal. So there's an incredible adaptation that has occurred where the animals are actually hosting these bacteria that are living, and the bacteria are obviously not harming them. They're helping them with this process of chemosynthesis, and they live together with an internal symbiont. Yeah. So it is quite remarkable.
It's so interesting to me. Now, is this the snail you were telling me about with the interesting name?
Yes. So, well, there's many different snails that I work on in this area, but there is one that has a really great name, and that's the genus name is Alvinaconcha, which is named after that submersible alvin that I mentioned earlier. But the species name is Strummeri, and that's named after Joe Strummer of The Clash, and is commonly known as the punk rock snail. And this is because these snails have a very spiky surface on the outside of their shells, similar to a punk rocker and their spikes. And so The person who named this particular species, Shannon Johnson, who's a Monterey Bay Aquarium Research Institute scientist, chose this name because of the spikes and the relation to sort of the look of punk rockers. So I do think that's a great story about these particular snails.
And it shows once again that science is a human profession.
Yes.
And it isn't necessarily some silly in-joke. It can be just whatever you're interested in. And I just love it. So thank you for sharing that.
Yeah.
So this is a wonderful situation. So when you study this, what are you measuring and how do you measure it?
Yeah. So these animals and their microbial symbionts live incredibly deep on the seafloor, sometimes I said kilometers deep. And so they're used to and adapted to extremely high pressures. And so what that means is that it's very hard for us to study them alive. It's not impossible. We can sometimes bring them up and keep them in these specialized high pressure aquariums that we can then expose them to different conditions and do actual experiments on them. But this, as you can imagine, is quite challenging technically and is often limited when we can, we could do it for the few weeks that we're at sea at a time. And because they don't serve, we can't bring them back to our labs and keep them alive or anything like that. So most of my work has focused on using samples of tissue from these animals that we collect and doing genetic work to understand these symbioses. And so lately, a lot of work in the lab has been trying to understand the diversity of different types of bacteria that associate with these deep sea animals, either within a region or globally across the world. And we use genetics and genomics to do that. And to understand how these bacteria might help an animal adapt to the specific environment of the vent or sometimes another type of environment that they're found at. And so we're looking at how different vents vary over space and over time in terms of the chemistry, other conditions, and how the bacteria help the animals cope with that.
I have an odd question, if you don't mind.
Oh, sure.
We've talked a little bit on this podcast about some of the tube worms found around hydrothermal vents. And in fact, these tube worms actually have hemoglobin related to our own.
Yes.
But hydrogen sulfide, even at the levels of, say, 50 millimolar that you often find in the hydrothermal vent fluid, that's super toxic to hemoglobin. So the tube worm hemoglobin is resistant to hydrogen sulfide. Once again, evolution is doing everything. Are there similar sensitivities the snails have to the hydrogen sulfide conditions?
So you're right that hydrogen sulfide is typically considered an extremely toxic compound for most things on Earth that breathe oxygen. It binds to the cytochromes that are the molecules and proteins that help us breathe oxygen, and then can inactivate them. And so that's toxic to things that breathe oxygen. So it would be toxic to these snails that are in fact breathing oxygen, even at the bottom of the ocean. So we don't fully understand exactly how they're coping with this. There's, to my knowledge, no hemoglobin that's binding sulfide in their blood. So yeah, we don't really have a great sense of the mechanism, but they certainly are bathed in extremely high concentrations of sulfide. It's possible that the symbionts that they are partnering with do help them in some ways with this, because the symbionts, as they go through chemosynthesis, are turning that toxic sulfide into non-toxic sulfate. But we don't really think that's the primary reason why they have the symbiosis. Really, the reason is because it provides them food, but maybe a byproduct of that is that it helps them deal with the toxicity of the sulfide that's around them.
And it's important to emphasize for listeners and viewers kind of the oasis that you described surrounding these hydrothermal vents. I'm not going to call the areas on the oceans. I was going to say the opposite of surface. That's the problem. But the sediment at the bottom, I'm not going to say it's lifeless, but it's certainly very low in nutrients compared to other places. And then when oceanographers began to explore these hydrothermal vents in the 1970s, they discovered this huge amount of biomass that was there, growing very quickly and very large.
Yes.
And so this must be just rife with competition within there. And I know there are other species that hang out there feeding off some of the organisms that are able to get their nutrition from hydrogen sulfide and CO2. So it must be amazing to see this diversity and large amount of life there at the bottom.
Yeah, absolutely. You said the word oases. That's often the term that people use for these because, as you said, the rest of the seafloor is not barren of life by any means, but there's not a lot of food there, which means you just don't have these kinds of abundances that you would see at these hydrothermal vents or cold seeps, which can rival, what we see in coral reefs or intertidal areas with the numbers and sort of biomass of organisms and animals that we see there. And certainly we at vents and seeps worldwide where these chemicals are available, we have lots of these dominant symbioses between animals and bacteria, like the tube worms you mentioned, the snails and mussels that I work on. There's lots of different types of other types of tube worms and other snails and mussels. There's also crustaceans like shrimp and the nicely named yeti crab. All of these animals have evolved to partner with chemosynthetic microbes to provide their food. And they are usually the dominant sort of foundation species at each of these vent or seep sites around the world. But as you mentioned, there's also all sorts of other life there too. There's free-living microbes. For example, there's lots of beautiful microbial mats that are living on the surfaces of the vents or the seafloor around, and they can be all different colors and extremely dense mats. And then there are animals that feed on the symbiotic animals or feed on the mats directly, like, some other crustaceans that graze. We see fish. So yes, it's this amazing abundance of life congregated together in these small areas on the seafloor because of microbial activity and chemosynthesis.
To me, and I know this is kind of a beginner kind of comment, but I love it so much. I keep forgetting it's actually dark down there.
Yes.
And so to see this incredible diversity and abundance of life in the dark is just amazing to me. I know some people have sometimes said, oh, this is a complete ecosystem that has nothing to do with sunlight. And that's not really true because oxygen is being used and oxygen is created through photolysis by the typical kinds of photosynthetic organisms we're used to, but it is a very unusual and fascinating system. May I ask one more question about the snails?
Oh, sure.
I have symbiosis on the brain, and I always have. So what about how those microbes are transmitted to the next generation? One choice is that they're transmitted through, say, eggs. The other is they have to be reinfected from the environment. Do you know the situation with your snails?
Yes, that's right. When you're in a symbiosis with a microbe and it's crucial to your life, to the ability for an animal to survive and persist, then you can imagine that these animals have evolved some mechanism where they can transfer their symbiont from one generation to another to make sure that their children are set up with their symbiont to survive. And one way of doing that is a direct transfer, as you said, through the eggs. We sometimes call that vertical transmission. And that does occur at deep sea hydrothermal vents, but rarely. The more common transmission system, as you also mentioned, is sometimes what we call environmental or horizontal transmission. And this is where the bacteria that are in these host animals leaves the animal or is expelled by the animal into the environment and then persists in the environment and is eventually taken up by the young at some point in their development, commonly in their maybe late larval stage or early juvenile stage. And in fact, we don't usually know for most of these animals exactly when and how they get their symbionts. But this has been a focus of my lab, is trying to understand how these symbionts are transmitted from generation to generation, and where the symbionts are in the environment, how abundant they are, because we know almost nothing about that stage of the symbiosis.
It's so interesting to me, and I did my graduate work on root nodulation bacteria, and the conventional wisdom is that when the plant dies, the nodules break apart and release the bacteria into the soil. So they act as a refugium to provide them. And I think that kind of depends on the association among the leguminous plants. But I love this idea of broadcasting it because it implies that there are mechanisms to recognize friend versus foe.
Yes.
And that interests me. Again, the difficulty is how to study it. I have heard, you're the expert, I have heard there's evidence for the tube worm's larval form having a specific type of aperture early that goes away when it's older. So during that period, they're colonized. But I don't do that work myself. I have heard that. So in a similar way, you have to consider how these things are being recognized by the snail. And I don't know how you'd approach it in an organism that lives so deep.
Yeah, it's very challenging. There has been some work, as you mentioned, on the giant tube worm and people studying that with what I think I understand are collected juveniles, from the environment that they were able to visualize the symbiont beginning to infect the animal in its juvenile stage. And we are trying to do some of that with the snails and mussels that I'm working on, trying to capture these earlier stages and try to visualize using microscopy or molecular tools to try to actually see the symbiont in the tissues and understand that. But, unfortunately, we can't, culture these animals in the lab. We can't culture the bacteria separately. And so doing the kinds of experiments that you're describing to really find mechanism is quite challenging.
Can you please, this is a totally Mark Martin question. What are the closest relatives of the endosymbiotic sulfur oxidizers that are living inside the snail tissue?
Yeah, so there's many different types. So that's the first thing, is that there's not just one type of bacteria. There's many different types from many different lineages. And so that's why it's a very interesting system for me, is to try to understand why partner with these different types of bacteria. One species might have one snail species might have a different one, and what does that mean for the host's ecology or evolution? But most of these chemosynthetic symbionts are gamma proteobacteria from a variety of different lineages. Many of them don't have very close relatives. Like they've sort of branched more deeply away from other gamma proteobacteria, and now we have, and and their closest relatives are other symbionts of chemoautotrophic symbionts. I do work on one species of Alvinaconcha that associates with either a sulphuromonas or a sulphurovum, which are Campylobacter, and those are unusual in that they do have close relatives that are in that same genus, the sulphuromonas or sulphurovum. That's unusual, though. Usually the symbionts are kind of quite different and divergent from other known bacteria.
So getting back to your idea of having corn living inside of us when we're next to a window.
Yeah.
It sounds like your snails are able to have a number of different plants become part of it. You know what I'm saying? It doesn't sound like there's a specific sort. Like if you look at rhizobium and leguminous plants, a particular species is generally what is able to form that association. But it's broader than that when you're talking about your snails, which fascinates the you-know-what out of me.
Yeah, it's generally specific. I didn't say this clearly, but it's generally specific where each snail species has only one, but sometimes two types of bacterial species that it can associate with. So, we do see some variation across different host species where even, these two types of Alvinaconcha, they're sister species in the same genus, and they have entirely different types of bacteria. Sometimes they even live together in the same vent habitat, these two species, and they each have their own bacteria. So clearly there is some specificity between the bacteria and the host. But what we have seen is that there is strain level variation. So different strains within a particular bacterial species. So if you look, for example, at one species of snail across its different geographic range at different vent sites, it seems to have partnered with strains that are very locally specific to that area. And we believe that has to do with this idea that when you are an animal sending your larvae out into the world without their symbiont, that's obviously a very risky proposition. You're sending your children out into the world without the partner that they would absolutely need to survive. But the risk of this is outweighed by the opportunity to then arrive at a site and take up a symbiont from that particular site, a strain that is then specially well adapted to that particular site and well suited to that site. And that's sort of the benefit that then outweighs the potential risk of sending these larvae out without their partner.
About 1,000,000 years ago now, I knew a woman, Debbie Millikan, who did a little bit of work with the bacteria that live inside tube worms.
Mm-hmm.
I'm just going to call them the Riftia symbionts.
Yep.
And she reported that bacterium had functional chemotaxis genes and functional flagellar mechanisms,
Mm-hmm.
which suggests that it's able to swim around when it's looking for a host. And
Mm-hmm.
these are the things that I'm hoping you're finding in your symbionts in your snail, that they clearly have a lifestyle outside the snail. We know they have to, but
Yeah.
it's interesting because you normally would think about things getting, as the symbiosis progresses, more and more Darwinoed, for lack of a better term. But that's not what we're seeing. And it has to do with dispersal, I would imagine.
Yes, absolutely. And so, yes, we do sometimes, although not always, see genes for motility in these symbionts, which imply that they could be, as you said, moving around and looking for their symbionts or looking for their hosts. We do also often see genes for heterotrophy in these symbionts as well. So again, implying that maybe when they're outside of their host, there might be opportunity for them to be taking up compounds that are available to them, and in addition to doing autotrophy. So my hypothesis is that they are probably not just persisting when they're outside of the host, meaning, staying in a static form and not growing, but I suspect that they are likely growing and actually able to divide and persist outside of their host, and that is part of their life cycle and lifestyle.
I recognize that recently there was a type of an alga that has a nitrosome. In other words, a bacterium that had been Darwinoed down to being essentially an organelle on the path to becoming the equivalent of a mitochondrion or a chloroplast. So, it makes you wonder why that hasn't happened with these thiotropic associations. Maybe they have and they haven't been found yet. Hard to say, but how fascinating that they're able to live outside the host so effectively.
Yeah, absolutely. I think it goes back to what I was saying before about the trade-offs in the system to the horizontal transmission, where you have, the symbionts living in the environment, and they need to retain the functionality to do that. They can't sort of become just an organelle. They can't become an organelle because they also need to be able to live in the environment. And I think that might not just be something that hasn't happened yet, but that this is actually an adaptation to the environment that these symbioses are experiencing, where they have these vents that are separated by long distances. The vents vary in their chemistry. Even a single vent can change in its chemistry over time. And so you would imagine that if you're going to try to make your children more successful, you would want them to partner with the best possible partner. And that, is not just by giving them the one that worked for you, but it's by sending them out to find a partner that's specific to the area that the children end up in.
I think that that's very well stated. And I was always surprised to learn the hydrothermal vents were relatively short-lived, and then other ones pop up. So that issue of how dispersal works is one that all of these organisms that take advantage of that particular lifestyle have to deal with.
Yes. Yeah. I mean, many vents are short-lived, like you said, but that also might be a bias of the ones that we've been most able to study. So the vents that are most well studied, at least by US scientists, are vents in the Eastern Pacific. And those vents do have sort of a high turnover rate. They have lots of sort of recent or sort of more frequent eruptions that then sort of change the vents, or maybe even pave them over completely. And then the vents have to start again, and the animals and the ecosystems there have to start again. In fact, in the news just earlier this year, some American scientists discovered that some vents at the Eastern Pacific Rise recently erupted. And so this process starts anew. And that's usually at the decadal scale, that we can actually observe this. But vents in other parts of the world are much more long-lived and may not have that type of disruption or disturbance. over short time scales. Like the vents in the Western Pacific have been very stable over much longer periods of time. And in fact, some of my colleagues have done studies over 10 years or longer where they go back to the exact same vent, and they look at it with the exact same angle, and they see that the animals are sort of in the exact same pattern over many, many years. Like they see the same, we don't know for sure if it's the exact same snails themselves, but there are always snails at this one part of the chimney, and there's always mussels at this other part of the chimney, and that hasn't changed for 10 years.
That's just amazing.
Yeah.
So talking about that reminds me of something that you discussed a little bit before the show began, and that has to do with Tonga and has to do with volcanoes. And I think that's quite a story if you'd care to share it.
Oh, absolutely. So yes, I've been working at hydrothermal vents in the Tonga region for many years, and in 2022 happened to visit these vents again, only a few months after a very large eruption occurred in that area, which your listeners and viewers may be familiar with. It's the Hunga Tonga volcano that erupted in January of 2022. It's one of the sort of largest volcanic eruptions I think we as humans are aware of. It shot material into the stratosphere. It was one of the largest noises ever recorded, and it made a huge impact in the region. Obviously, it had some effects on the people of Tonga and the island of Tonga, and unfortunately, there were some people killed during this eruption. But it also, of course, infected the marine ecosystem around it as well. And so we happened to be on a research cruise that was completely unrelated to this eruption a few months after. We had been scheduled to go at this time anyways. And we ended up out there in sort of March and April of 2022. And we were at these hydrothermal vents that are about 100 kilometers from the volcano. And we did not expect to find any impact of the eruption. We had talked to volcanologists before going. We had, sort of gone through the possibilities and were assured that we would have no, problems, so to speak, of going out there and continuing to do the work that we had planned. But when we reached these sites, what we found is that many of them were covered in these really thick blankets of ash from the volcanic eruption, sometimes up to over a meter deep of ash. It was sort of like pillowing snow all over the seafloor. And so we dove with the ROV to the seafloor on one of our first dives and discovered this landscape covered in ash. And then as we explored the vent fields, found that most of the animals at this vent field had been killed by the ash that had fallen on them. And so the communities there were entirely devastated by this eruption. And so our research plan and our project had to quickly pivot to an exploration of this disturbance event and what it would mean for these ecosystems. And so we were able to sample some of the ash directly and take that back to, eventually to the lab and study it. And then we also were able to characterize how this ash impacted the animal communities. And so before the eruption, there was a French expedition out in 2019, which is the most recent expedition before ours. And they, documented these sites clearly showing no ash. And then we were back there in 2022, right after this eruption, and, found is that these landscapes covered in ash and that this, completely decimated the animal communities there. We had some sites that went from a normal, abundant animal cover to almost none of these animals seen alive at all. So it was a really big impact.
That's just amazing. And, I think people may have seen in the past what happens when you get an eruption in the ocean that creates a new island. And then I remember seeing all these reports of how life arrives there. But you're talking about stuff at the bottom. And
yes.
Do you have any idea how long it'll take to recover from this?
No, and that's one of the things that we'll be working on in the future. I have been lucky enough to be funded to go back there again in the future. We're waiting to get on the schedule for the next cruise, and we'll hopefully be able to follow up on this. As I mentioned before, these parts of these vents in this part of the world had been thought to be fairly stable and not experience a lot of change or disturbance naturally. And so this is we think is an unusual event for them. And so we don't really know how they'll recover. The other aspect of this that's different than what we've seen at other vent fields is that this is, as far as I know, the first observation of ash cover on these types of animal communities in the deep sea, as opposed to other eruptions where you have lava coming out at the seafloor and it essentially paves over the communities there with a hard, lava rock eventually. This is ash, which is basically like sediment. It's soft, like silt, sand kind of material. And so that has settled over everything. And these animals, we don't think are adapted to living or reproducing with their, with their young being able to settle on sediment. They're used to hard rock. And so what we don't know is even if they're surviving animals that can reproduce and they can send their, the larvae out to recolonize, it may mean that they may not be able to actually survive and be successful in an environment where there's now basically sediment everywhere instead of hard rock.
That is just amazing. It's funny because and for all we know, there'll be microbial communities that are well adapted to living in silt that will come to predominate there, I would guess.
Yes.
But I also don't know the physical effects of the silt. And by that, I mean chemically. It might change the pH in some fashion, it would occur to me.
Yes, exactly. So we are working with chemists to be able to understand that and predict that. And that is, one of the things that might happen is that you could see changes in the chemistry as the ash interacts with the chemicals coming out in the venting fluid. You might see more or less of the chemicals that these animals or other chemosynthetic microbes need to survive in the system. I will say, though, that when we were there, even though, the animal communities were totally devastated by this ash, we did see some actively, growing microbial mats on the surface of the ash. So there are definitely some microbes that will persist, either on or in the ash, but the animals themselves we are unsure about.
I have a saying, Roxanne, about the microbial world: first evolved, last extinct.
Yeah, absolutely.
And it's really true.
Yeah.
How wonderful that you get to see what I would imagine would be a laboratory for how a new site would develop. Although it's not the same as the sites that you studied because of the silt, you're now seeing the colonization by microbial mats, and then hopefully animals, creatures of different kinds will begin to inhabit it.
Yes. Yeah. And we're also very interested in trying to understand if the symbionts have remained in the environment, even though maybe the animals haven't died or have died, that their symbionts may be hanging around and what that might mean for their ability to recolonize. We have some work that's not yet been published where we've actually looked at symbionts in the environment at these sites. And we do, in fact, we can detect the symbionts in the environment, in the water or on the surfaces of rocks in this environment, even after the eruption. What we don't know is how well they're going to persist in the future, if they're actually even still alive. These are all, questions we can't fully answer. But the good news is the symbionts at least are still detectable. So hopefully that means that they're still out there able to be waiting for animals to come and partner with them.
I think it is such an exciting thing to look at. It's a wonderful combination of field work in some, and when I say exotic, I again am well aware of the challenges of field work on ships, but so I'm told anyway. Yeah. I haven't done it myself. And the bench work, and I know in some cases you have the equivalent of research benches in a ship as well, which has its own set of challenges on top of that. Yeah. But what a wonderful Venn diagram when you think about it for anybody that would think about being a particular microbial scientist of the world.
Yeah.
And what better system to look at than these, I'm going to call them the primary productivity that we see around hydrothermal vents, because that is kind of what you're looking at, rather than driven by sunlight, driven by the oxidation of hydrogen sulfide. Fascinating beyond belief.
Yeah, it's absolutely primary production. And as you said, it's an incredible opportunity for, people interested in both, sort of traditional microbiology or microbial ecology. And then if you are also interested in, going out and doing these exciting expeditions, which, as you said, are a lot of work. They are a lot of hard work. We're often working around the clock when we're out there, but they, are amazing opportunities, that are really unique and incredible. So I do feel pretty lucky to be in this field.
As we start to kind of wind up our time together today, there were two things I was going to ask you to do if you could. The first would be talk a little bit about your path to the microbial sciences. And this is because over and over again, I've had students say to me things that make me believe that they think, using a lot of modifiers, it appears that many students believe there's only one path to the microbial sciences. And if nothing else from this podcast, just as the people are diverse, so are their paths. That's thing number one. And then before we sign off, I would like you to be able to share maybe 3 lessons that you would like every listener and viewer to take away from this session.
Okay. My path to microbial ecology and microbiology started fairly early. I was one of those weird children that got exposed to microbiology very young and suddenly knew that that was a field I was interested in. I think I was around 10 years old when I participated in an after-school program in my town where a local biotech company came and did activities with kids. And in that program, we were able to actually look at microbes under the microscope, and we were given agar plates, and we were able to rub whatever we wanted on those plates and see what grew. And I was just enthralled. I loved this sort of invisible world that was around us, and knew that was something I really wanted to study. But I was only really aware of microbes as pathogens, and that's what I thought I might be interested in the beginning. And so going into college, I thought I was interested in pathogenic microbiology or biomedical microbiology. And when I got to college, I became exposed to the idea that there's this whole world of environmental microbial ecology out there, people who are studying microbes in the environment. And so through that, I discovered a love for microbes in the marine environment, and also became very interested in how microbes impact ecosystems. And microbes impact every ecosystem on Earth in so many ways. But for me, they're finding this really strong link between microbes and some specific organism, either an animal or a plant or something, where you can actually see their really strong and direct effects, like through these symbioses, is really what sort of I came down to. I became very interested in this idea that microbes could have this profound effect on life through a symbiotic interaction. And so I actually ended up working in these deep sea symbioses, not through a sort of love of the deep sea and sort of expeditions and the exploration and discovery aspect, which many people who work in the deep sea world, that is sort of where they come into the science through. But for me, it actually came in through the symbioses. I was very interested in symbiotic relationships, and these symbiotic relationships that in the deep sea are really just amazing, partnerships that sort of provide a really great way to study how microbes can impact, other types of organisms.
I didn't have opportunities as beautiful as those, but there were things like that for me starting early. And that's such an important thing to make available to people. I'm glad you had that opportunity. I'm glad I did. My life would be completely different without those middle school, after school programs.
Right. Yes, exactly. I mean, this definitely just is one example of how these types of things can make a difference, and that, even early on, you might be able to capture someone's attention that could affect their whole life going forward.
Absolutely. Absolutely. I mean, by the way, for me, one of my brother's friends was an amateur scientist, and he gave me a streak plate with luminous bacteria. I was 10.
Yeah. Okay, great.
Yeah. Of course, and then that, I was like Saul of Tarsus. I was struck by this microbial viewpoint that I had to follow up on. But then I want to be a microbial evangelist. So there you go.
Yeah. That's great. Wonderful.
So what maybe 3 things you'd like everyone to take away from today's session?
Sure. I guess first would be about chemosynthesis and understanding that as a process that is important for life on the planet, but maybe one that not everyone is very familiar with, that there's, not just photosynthesis as a way to sort of create the building blocks for life. There's also this other process, chemosynthesis, which uses chemical energy. I would say that's one. I would also say that, that there's these sort of partnerships between organisms like animals and bacteria that are critical for the life of those animals. And so this is the ones that I study in the deep sea are these really interesting examples. But as I'm sure, and I'm sure many of your listeners and people know, is that, that microbes affect pretty much all organisms on Earth in some way, often through symbioses. And so these deep sea symbioses are just one of those examples. And then the last would be that if you're interested in microbiology and also doing exciting fieldwork, that there are definitely ways to partner those two things in a symbiosis and become, a field-going oceanographic microbiologist who has opportunities to study these extreme, remote environments, but then also focus on microbes as well.
I think you put it very well. And it's funny because we all get pushed into particular areas. I was constantly asked when I was in college why I wasn't interested in studying cancer, which you understand. But as I used to say, my microbial heart wants what my microbial heart wants.
Yes, absolutely. Yes.
So finding what...
I was going to say, I think that, people often, as I said, think of microbiology and they think pathogens and they think, harmful microbes. And of course, that's not the only microbes on Earth. In fact, those are probably some of the smallest numbers of microbes. Most of the microbes on Earth are not harmful and often are helpful.
It has been a wonderful session, and I've really enjoyed this opportunity to talk with you. I believe I've been to one of your seminars at ASM in the past, and I can't wait to actually shake your hand in person at some upcoming meeting.
Yes, absolutely. I would love that. Thank you so much for having me. I really enjoyed speaking with you.
I wish you and yours the very best.
Thanks so much.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send suggestions, questions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, as always with epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Roxanne Beinart can be found in the Graduate School of Oceanography at the University of Rhode Island in South Kingstown, Rhode Island. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile every time I hear it. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.